In this video, we will be learning about mitosis and meiosis. Basically, our focus in this video will be for mitosis. So, mitosis generally takes place in the vegetative cells, whereas meiosis takes place in the germ-line cells. So, in the cell cycle, whether it's a mitosis or meiosis, there are four phases which takes place, and any cell who has to divide have to undergo in these four phases. First is G1, then S, G2 and M mitotic phase. So in the G1 phase, basically when the stimulus comes to the cell for the division, then it will start synthesizing the organelles and the proteins which are required for the replication and for the division of the cell. So that takes place in the G1. Then comes the synthetic phase. In synthetic phase, basically the replication takes place and the genetic material, which is here if 23 pair of chromosomes, so it will increase to 46. So in this synthetic phase, basically the replication will take place, the ORI formation will be there, and the DNA polymerase will synthesize a new copy of genetic material. Then the G2 phase comes. In G2 phase, basically the cell will increase its size so that it can equally divide into two parts. Then after the G2, M phase comes. This is a mitotic phase. So mitotic phase is, in the case of mitosis, is divided in four subphases. First is prophase, metaphase, anaphase, telophase, and then comes the cytokinesis, which is not the part of mitotic phase, it is a part of the cell cycle, G1 basically. So in the prophase, first of all, what happens? This is a normal cell and this is the chromatin genetic material. So in the prophase, the nuclear envelope will get dissolved slowly, and also the chromatins will be condensed. So after condensing, it will look like this. So after condensation, it will look like this, and in the prophase, the condensation of chromatin and the disappearance of the nuclear envelope will take place. Also, the centriole will get divided and will start moving towards the opposite pole of the cell. Like here, these two are the centrioles, which are moving to the two different poles, and these are the condensed chromatin fibers, which is a chromosome. And this is a small part is a chromatid. This is a chromatid. Here in the center, there is a centrosome. So in the prophase, this is the prophase where the condensation will take place. After that, the metaphase comes. In the metaphase, both the centriole will go to two opposite poles, and there the spindle fiber formation will take place, and these spindle fibers will join on the centromere position from both the sides, and these chromosomes will align at the center of the cell. This is also called the equatorial plate or cell plate, equatorial position. So this is the metaphase. So in this phase, all the chromosomes will align at one position, or we can say on one line at the center of the cell. After that, the anaphase comes. The microtubules which are attached to the centromere, they will pull the chromosomes apart and apart at two different poles. So here, as you can see, the microtubules are segregating the chromosomes, and they are moving towards the different pole at the anaphase. In the telophase, these condensed nucleotide will be changed into a chromatin, and this is a chromatin, and slowly the nuclear envelope formation will take place, and for the cytokinesis, slowly the plate formation will occur. This is a telophase. So at the telophase, the division of the genetic material or cell is almost complete, and the nuclear envelope formation has started. Then then there is also one phase, that is the cytokinesis. Cytokinesis means the division of the cytoplasm. So division of the cytoplasm takes place after the telophase. Here the both the cell will equally divide into two parts. So this division takes place by two methods. If in the plant cell, then it will takes place by, because plant cells contains the cell wall. So here the organelles, they will come together here, and this process will be a centrifugal. Because of the centrifugal force, it will means divide like this by this force. Whereas in the animal cell, because no plant cell wall is there, so this division will takes place by the centripetal force. So it will come the inwards, and there will be a formation of furrow. This is a furrow. So this is how the mitosis takes place in the cell.